Oh, dang. What's up, world?
All right. Hello, World Corona Chronicles. I got a very special guest here. And without further ado, this is Mike. I'll let him explain a little bit about his background. And you're going to learn something very informative today, for sure. What's up, Mike?
How you doing, Sterling? So I'm a classically trained virologist, and after that, I worked on HIV, human cytomegalovirus, and I've worked on a bunch of phages and those type of virions. Um, I went to grad school at Caltech for molecular biophysics, and now I'm working for I wouldn't call it a startup, but it's a small company, and we're making a detector currently for the SARS-CoV-2 virus. It's a near real-time detector. So,
That'll be useful. All right. So I guess the biggest thing is, what is a virus exactly, and what does it do to us?
okay, so that's a really good question. It's also a very difficult question because there are so many types of viruses out there. Their structures span, so many different kind of shapes and infectious pathways. And it's probably not the right way to put it, but um, so when you really get down to it, because some of them have an envelope, some of them don't have an envelope, most of them have some form of outer shell. They have binding receptors on the outside, and then they have a nucleic acid on the inside. So if you were going to ask what is a virus is generally a container, and that container contains a nucleic acid, either RNA or DNA, and it is able to infect a host cell and take over all of the host machinery, or at least what it needs, not all of it, take over the host machinery in the cell and replicate itself. And these things are incredibly efficient. They can take one human cell and turn them into virus factories that can make 10,000 virion particles that then they release into the bloodstream or back into the respiratory tract of the host, where they can then go and infect other people and infect other cells.
In this COVID-19, the corona SARS, or whatever you want to call it, do you know what this does to us? They say it's an upper respiratory infection. Is that due to the virus itself infecting us and causing this upper respiratory infection, or is this kind of like a catalyst to stimulate other infections to enact inside of us?
So just to get the naming straight, so we're on the same page, nCoV was the first name of the virus when it was still in Wuhan. And then the WHO changed it to be SARS-CoV-2, because it is a SARS virus. And then, but the disease it causes in you is called COVID-19. So the first one was nCoV-19, and then it became SARS-CoV-2, and the disease is COVID-19. Just to start that out with. So when I'm saying SARS-CoV-2, I'm actually talking about the virus itself. When I'm saying COVID-19, I'm actually talking about the disease. What it does to you, it's some of the best literature out there right now is on what it actually does to humans, because they're autopsies and dissections of humans. They're the clinical results from China, and there's the least amount of speculation in a lot of those papers. To start with, the virus infects it infects your upper respiratory tract, your throat, your mouth, the upper part, and then it goes through a serotypic change where then it migrates down into your lungs. And there's other pathways that some people have observed, but they're not well proven yet, and that is through your gastrointestinal system, where you actually consume the virus and swallow it, and it goes down into your stomach, and then that's why you get excreted particles in your feces. Also, by swallowing all the virions that are kind of rupturing out of your throat, you're swallowing a lot of those, which
And
leads
those
to
and
the
those
myth.
can infect you, or what do you about your stomach acid? How's that?
There are some papers that say that those that you can harvest live, not live, because as we said earlier, viruses aren't really alive. They're more like a packaging particle. They're just a package. So it's tough to say if viruses are alive. So what I'll be talking about whenever I say it is viruses have the ability to infect, right? So if you have feces that's able to infect, right, that means there's active, infectious virus particles there. And there are some papers saying that, then there's other papers that saying there aren't. You got to remember, we're in kind of a fog of war situation right now where there's so much information coming out, oftentimes contradictory because it's non-peer reviewed. It's amazing when this is an unprecedented time in science, but at the same time, it comes with, got to take everything with a grain of salt.
Right. So it infects you somehow. This virus gets inside of you. The envelope attaches to,
Mm-hmm.
or the envelope attaches to some cell. In this case, it needs your, correct me if I'm wrong, it needs a specific cell inside your lungs. It attaches to this and then just injects it with its DNA material. And then from there, it just kind of, it's almost like it lays eggs inside your cell. And then it just, breeds these viruses and explodes, and then it just gets everywhere.
That's not too far off. It's a pretty good way to do it. So what it has is the virion particle has a spike protein. It's called the S spike protein, right? And that's my finger sticking up here. And what that does is that is a receptor for the host cell. And only certain cells in your body have the receptor that binds to, and one of the receptors is called ACE2. And I'll get into that in 1 second. But so you have your host cell and then your tiny little virus. I mean, the virus is, host cell is giant and a virus is a tiny little particle. It's like 100 nanometers across versus a host cell is about 15 microns across. So it's a big difference, 150 times bigger in terms of length, but in terms of volume, it's infinite. Not really, but you know what I mean. So what happens is that S1 protein binds to your cell in your mouth. You pick it up by your hand, you touch your face, you swallow it down into your throat. It floats around in the air, comes into your lungs, gets stuck in your mouth, and it binds to your epithelial cells inside of your mouth. And so the receptor binds, and then there's this event where and these are enveloped viruses, which means they have this outer fatty layer right underneath the S1 protein. That envelope and your envelope fuse together. And when it does that, it releases the positive strand RNA from the virus into your cell. Your body then just takes that and starts producing the proteins on that positive strand. And what then happens is that the virus encodes for certain proteins, like RNA-dependent RNA polymerase, right? That's one of the big subjects right now in this virus. And so that polymerase then is able to take its own first DNA strand and then pump out a whole bunch more DNA strands. So, and then each one, RNA, I said that wrong. It takes its own positive strand RNA and pumps out more RNA, a whole bunch of copies of that RNA. And then, so once it starts pumping out the RNA, it starts making more protein, turns your whole cell into this excretory pathway where it sends it to the Golgi apparatus, and then it kind of buds out of the cells. And it just pumps these virions right out the surface of the virus of your own cells, which then they wash down to other tissue or other areas in your throat and deep in your lungs, which is the second phase of kind of the infection. That's when it gets bad, is when it migrates into deep lung tissue.
So it's already replicating itself in your throat before it enters your lungs.
Yes.
So this theory of someone sent me an article about drinking water. You can wash it, just drink every 15 minutes, drink a couple cups of water and wash this thing down.
It actually may delay your serious symptoms, because let's say you can prevent 80 or 90% of the virus load from making it down into your deep lung. I'm not saying this is a solution, and don't drink hot water, don't drink bleach, don't drink methanol by all means. We laugh, but what is it, 2 days ago, like March 27th or something, 400 Iranians died from drinking methanol because they were told that it washes it out of your. It's a serious thing. People are buying so many different things right now, but there may be something to that where you can simply slow the virus progression enough for your immune system to kind of catch up, because your immune system takes 4 to 7 days to start making, for your B cells to turn into plasma cells and start making antibodies to start fighting this.
Right. So how long, speaking of that, how long does it take then? I guess the correct wordage would be, what's the incubation period from once the virus gets inside of you until you start having the effects of fever or side effects? What are they saying about that?
Well, you may never get them, to be honest. You generally have some form of bilateral pneumonia, even if you're very asymptomatic. Once it kind of gets really ripping into your body, you could be asymptomatic and kind of have a walking pneumonia. But the general time, what they say is it's centered around 5 days. The peak is 5 days, and it's generally one standard deviation on either side of that. So 95% of the people will have it between 4 days and 7 days. It might be 6 days is the center. It's 5 or 6 days is the center. It's 4 to 5 days at the earliest, and roughly 8 to 9 at the latest of that main group. And then there's rare cases that go on either side of that, outside of the normal standard deviation.
And then the extreme cases, obviously, of the asymptomatic, that don't show anything for months on end.
Mm-hmm.
And I heard children as well. There's this children. One of the bigger problems that I've read about was influenza and viruses like this. They actually infect children, but children won't even show symptoms, or rather their immune system was so strong that they would actually fight it without even having the fever, without even having the symptoms. But the problem is they would spread it then to their parents.
Mm-hmm. Yeah. That was a really big puzzle that Wuhan was kind of discovering early on, is why are there no children showing up in the hospitals? So there's papers out right now that show that what the Chinese originally thought was that the virus was not infecting the children because the parents were protecting them and keeping them isolated and doing a job. But the reality is these children were probably just the asymptomatic carriers of the disease. So they just seemed to have less major symptoms, if that makes sense. It just doesn't do as much to them.
And how is this one so much different than the influenza, the flu, the standard flu? There were so many, I don't want to say propaganda, but people were posting things of how this is nothing compared to the flu. The flu kills so many people a year, and this is nothing compared to it. How does this virus stack up next to the flu, and why is it so much more dangerous? Why is it something that we're so concerned about as a country and as the world?
Oh, that's a week-long topic right there in itself. But it needs to be addressed, and I think other people are addressing this to a really good degree. But the bottom line is people were saying that it's like the flu, because we all think the flu is harmless. For the majority of us, it is. It's fairly harmless, and we have this idea that, oh, the flu's not deadly. Then they would kind of sneakily transfer that to the Spanish 1918 flu, which was basically 101 years ago, right? And that was serious. When you read up some of those reports on that, that was devastatingly bad to the world. I mean, I think 500,000 people died from that. That was a serious thing. Like Trump's uncle or grandfather or something died. I mean, so many people in the world died from that. Why is this more dangerous? A number of things. Number one, there's no herd immunity. No one's had this virus. When you're calculating what's called the R0 or the R0, which is how contagious a virus is or how easy it is to spread from person to person, R0 is one of your biggest factors. Because if you have, like measles, has an R0 of 12, okay, which means each person infects 12 other people, okay. So if you didn't have all the vaccinations against measles would be incredibly contagious and rip through the population like crazy. But we have what's called herd immunity to measles, and that's why it doesn't do that. This virus, there is no herd immunity, and that's going to be one of the biggest problems is as we transition from this stay-at-home work. This is my lab I'm in right now at home. We transition from this stay-at-home type of mentality to re-venturing back into society. If there's not a herd immunity, we have to be employing these non-pharmacological interventions. And if we're not doing that, it's just going to pop right back up. Now, do you want me to keep going on what's different about this and influenza, or that's the biggest thing?
What do you mean pop right back up? So as we know, influenza, the common flu, it does what you're saying, it pops right back up. But what do you mean pop right back up in the sense of not having herd immunity?
So if I don't have it, right, if I haven't gotten the disease and I go back out in public, I'm now susceptible to it. And somebody who has it can then just restart this pandemic all over again. Because right now, a very, very small fraction, it's like, I don't even know the percentages, but it's a few hundred out of every million people that have this virus. Less than a 0.001% of the population has the virus right now. And until you have a sizable amount of the population who has the virus, who have had the virus or have a vaccine, there is no herd immunity, and the epidemic can just start right back up. One person will spread it to 2.6 or 3.3 people, whatever you believe the number is, and then that will just exponentials are very fast to take over at that point.
And how do we get this? So I heard at the beginning it was a snake, then it was a bat, and now it's some antelope-looking animal, armadillo-looking armored animal that they found in the Wuhan market. How does a virus actually like this jump from one animal to the other animal, and then eventually into us?
Yeah, it's a good question. This is a whole realm of virology called zoonotic viruses, and zoo is like zoology or like a zoo,
Yeah.
right? It's animal, and so these are zoonotic viruses, and what they do is they have a reservoir. Generally, they live in a virus. They live in like the pangolin, like you were talking about, or a bat. And these, there are some viruses that freely jump right back and forth. There's like a grasshopper one that jumps to birds and jumps right back and forth. So some of these viruses just naturally jump right back and forth. If they have enough of the machinery that they need in both organisms, they can just hop right back and forth freely. There are other cases where you have what's called an emerging zoonotic disease, where it pops out of the animal reservoir and jumps into humans. And the truth is, these do this all the time. You don't even realize it, but you're breathing, I think the number is 100 million virus particles every day, right? Because you got to remember, there's viruses in the plant behind you. There's viruses going all over the place, but they're not infectious to you, right? Every species has their own viruses. You can, hopefully we get a chance to talk about the myxoma viruses in rabbits later, but all these different species have their own viruses, right? And so, but what happens is sometimes a virus can jump to another host, and it usually does so very inefficiently. You saw early on in this epidemic, pandemic, I don't know, whatever you want to call it, this pandemic, that what happened was some lady's dog in China became infected, and they were actually able to isolate RNA for the virus out of the dog. And this happens all the time. You're getting infected with viruses. They just don't go anywhere. They infect one cell. They can't use your own cells, polymerases. They can't use your cells, whatever it is that they need from that other host. And the infection doesn't go anywhere. It stops with your first round of cells. It might make it to your second round of cells. It might even come out of you and infect another person. But as it runs out of the material that it needs, it stops. And so that's what they were continuously looking for in the early days of this virus, is when the infection becomes sustainable with person-to-person transmission, not people going back and touching the cobras or the bats or whatever people were believed to have been getting it from early on, but whenever a human can transmit it to a human successfully numerous times, maybe 3 or 4 jumps, and at that point it's now our virus. You know, we've now taken that virus. It can probably jump right back to the pangolins or the bats at that point because it's still similar enough, but it is now a sustained transmission in humans.
When you're talking about these reservoirs, are we talking about feces, like fecal deposits? Are we talking about some kind of reservoir for contamination? What is that?
Oh, sure. I meant an animal reservoir is it's like a species or like a group of, it doesn't have to be one species. It can be a family of like primates, for example. It can be, it just means an animal reservoir. It means like a reservoir holds water, like these animal species hold this virus.
Got you.
Right? They have the ability to become, they have the ability to become infected and transmit it on to another thing.
So yeah, when they hold this virus inside of them, they're acting as the reservoir. Are they necessarily having the same symptoms we're having? Is it also causing damage to them? Or would it be, for instance, like my beautiful plant here infecting me and it dying from a certain virus, but because that virus doesn't necessarily, or I don't necessarily have the cells, it doesn't do anything to me?
Yes is the answer to all of that. It can have different symptoms. It can have the same symptoms. there was this one, I think Carl Strauss is working on it at Caltech, and the virus he was studying, don't hold me to this, please, but the virus infected, I think it was grasshoppers and birds, and I'd have to look up the numbers and the names, but it had two completely different symptoms in both of these animal reservoirs, or in both these animals, and I think it made one of them very
So it had different effects on each animal in particular.
Sure.
So like the
Just like
grasshoppers would have like a defective leg, whereas the birds might have respiratory infection.
Sure,
Something
sure.
like this.
And think about it this way, right? So you have a particular set of cells in your body that have receptors for this virus. And it can even infect, if you have cells that don't have that receptor, it can infect those generally. I mean, it may happen to, but it'll be very low probability of infection rate. And that brings up an interesting thing like this ACE2 receptor. The S1 protein binds to the ACE2 receptor. So what you can do is you can look, where all in the body, and they just released this study the other day, they look, where all in the body do you have this ACE2 receptor, right? Well, it turns out ACE2 is all over adipocytes. Adipocytes, you know this, are your fat cells, Dave. You don't have any, but the rest of us all do. And yeah, right. So, but the thing is a lot of things people are saying that this virus becomes compounded when you have a lot of fat, right? So obesity may be, and this is again early data, but obesity may be another one of these compounding factors that leads to increased rate of mortality in patients.
Right. Yeah, I remember at the beginning they were saying, for obvious reasons, asthma, smokers, and a couple other things. And that's one of the main worries, right? Is they were worried when it does come to America, it's not China, right? They don't necessarily have an obesity issue like we do here, but they had the elderly smoking and they had their own issues as well. But one of the main curious points was when it does come here, what is it going to do to our obese population, which is kind of scary. Another thing I want to touch back on a little bit was the herd immunity. There was an article, I have it here, and I'll publish it in the description section below. It talks about a 2007 study, obviously about the SARS, the first SARS-CoV-1 virus. And it talked about, in a way, herd immunity. It talked about our pathogen levels building up, our antibodies, or sorry, not pathogen levels, our antibodies building up against this first SARS-CoV-1 infection or virus. And it says 2 years, we carried a strong population of these antibodies which would be resistant to the virus, and then after the third year it started to deplete. Is that also something to worry about? I mean, if we do, like they say, 50, 60, 70% of the population get it before this thing could eventually eradicate itself based on herd immunity, is that something we might have to be worried about? Like, could we get infected again in 3 years?
Yeah, no, that's absolutely right. And so what they're doing is they're looking at what's called viral or antibody titer. So antibody titer, when I mention that, is just going to be how much antibody you have in your blood. And they can extract it out and just do an antibody ELISA, and they can actually quantify how much antibody you have in your blood. Or you can use like a viral epitope. And we can get into, this is one of the ways they're testing right now. You take a piece of the virion, you take a piece of that spike protein, and you then put it on there and flush your blood over it. Well, all the antibodies that you've made can stick to that particle. So then you can simply count those through some colorimetric assay or however you want to do it. And you can tell what your antibody or your viral or your antibody titer in your blood is at that point. And yeah, if it goes away in 2 to 3 years, like that paper you were just saying suggested, you may either have to get a booster every 2 years, or you might be able to be susceptible again, which is kind of when you get into what influenza was about, which is maybe it would have been a good idea for me to talk a little bit about that, why we get infected with influenza every couple of years. And part of it is that you lose your ability to fight the disease every 3 or so years. When you combine that with the fact that in influenza, there are a whole bunch of the H and N proteins, let's just call them that. Those are the surface proteins. And you have a whole bunch of different types. You have H1N1, that's the one everyone talks about. That's the Spanish flu. You have H4N4, you have basically H1 through I don't even know, I think it's 8 or 10. Somebody I'm sure will comment on that. I don't remember what the top range is, but it's the same with Ns. So you have a whole bunch of different combinations of that virus that's going around, all the different H's and all the different Ns. And when you combine that with your resistance to any one of those, it only lasts a couple years. That's why you can get sick with the flu every 4 years, even if you're getting vaccinated. That combined with the fact that they don't give you a vaccination for every strain.
So these multiple different H1N1, H1N6, whatever it may be, are these mutations? Would you call these mutations of the virus, to where the virus has a different spike protein on the end? So the H1N1, H1N6, for example, they're completely different, and you can't have a vaccine for obviously every combination of this kind of virus, right?
Yes, absolutely right. At
Okay.
one point, they were mutations. At one point, they absolutely were. When H1 became H2 from H1, it was a simple mutation or a number of mutations that resulted in that spread. And realistically, there are probably different H's that are in between there. Virus evolution, maybe we can talk about that a little bit later, is not like any other evolution you get into, because viruses don't breed with each other. Very rarely do they infect the same cell from two different virus strains, but they're always diverging. And the only thing keeping them together is natural selection. The pressure to bind to proteins, for example, on the host cell, or the how to make a better polymerase. There's only so many ways to make a good polymerase. And if you stray too far from that, well, then you're not going to make too good of a polymerase. So we can talk about that later if it's relevant.
Yeah, well, I think it might be relevant now, because there's another article, and this is more relevant than ever. And so sad for what's going on in New York. They're just being hit so hard, and we talked about this before, and with Chris, a brother. Because of their population density, New York was it was almost inevitable that something like this would happen. I mean, in walking around Times Square, I think it's something in the millions and millions of people pass by there every single day within a proximity of only a couple feet from each other. There's this in the city.New York. It was an article just posted talking about, and this is kind of scary if you ask me, but 26% of the patients in critical care were between the ages of 18 and 49. So I would like to touch on that. And also, from my understanding of viruses, they're actually supposed to mutate to keep us alive, because they need us to stay alive. So why would this be sending younger people to the hospital? And after, maybe we can touch on again how viruses actually mutate.
Okay, so one of the problems right now is that a lot of the elderly are, and not a problem, one of the great things right now is a lot of the elderly are sequestered in their homes. So they're becoming less contact with getting in less contact with the virus. These are these non-pharmacological interventions which are crucial to curbing this virus's growth and to actually reverse it. That's what China did. They basically sequestered everyone in their houses, and then the virus basically dropped to a level that the emergency rooms could keep up. And that's the whole strategy of this virus, right? The strategy of this virus is to simply simmer the burn rate down to a level that our hospitals don't become overwhelmed. Because right now it's 19%, based on some numbers, of the people who get this virus are hospitalized. And so the strategy right now is, if you can keep that rate low enough so the 19% who are hospitalized and require hospitalization can get hospitalization, you drastically reduce the number of total deaths. Because if everyone in America, for example, got this disease tomorrow, our hospitals would be overrun, and we would have millions and millions of people dead. But if we keep it burning at a slow rate, our hospitals can clear the virus, can clear the sick people out, and which is one of the big differences also between this virus and the flu, right? The flu hospitalizations take 4 to 5 days. This can take up to 4 weeks before you're discharged from the virus. Therefore, you're on a ventilator. You're not on a ventilator for 2 days in the hospital, 3 days in the hospital. You're taking up a ventilator for a good part of a month. That's a massive weight on the ventilators. So as you were saying, that viruses tend to keep hosts alive. One of the things about evolution is there is no kind of, there's only hindsight when you talk about things like that. The virus does want to keep you alive. You are absolutely correct in the sense that it wants to keep you alive so it can spread faster. Because if I get infected with virus 1 and you get infected with virus 2, and let's say we're twins, right?
Almost,
Then,
but yeah.
sure. If if your virus keeps you alive longer and spreads more, well, it's going to have a better Darwinian fitness, right? The longer it can keep you alive, and actually the less your symptoms are too, right? You can continue to go about your normal ordinary lifestyle. And if you can go about your normal ordinary lifestyle, you can increase that R naught number that we were talking about, the infection rate. And so instead of infecting 2 people, you're going to infect 4. Well, pretty soon my virus that I have that killed me, right, is going to basically die out of the population. Yours is going to be the predominant strain in the population. So it doesn't want to keep you alive so much as it wants to spread more, right? And this is a particular case that they've proven in some of these real world examples. One of the cases, I get a little excited sometimes, and I'm not going to get excited because this is a very serious issue. But there's this case so what they did in the 1800s, 18th century, something, I know that's 100 years apart, but I don't remember the dates when they introduced rabbits to Australia. You spent some time in Australia, so this may be relevant for you. Well, they introduced rabbits to Australia, I think it was 18th century. And what happened was these rabbits have no natural predators there, and they took over the whole island, continent, whatever you want to say, right? And so in the 50s, because once you introduce a few rabbits, right, rabbits do what they do, is they breed like rabbits, and they just swoop around the whole place. So what they did is they introduced this virus called a myxoma virus. And myxoma virus is a pox virus. It's a really horrible virus. The rabbits end up with these large pustules that bleed, and it's a really disgusting way to die. And it was like, I think 98% lethal. Don't hold me to that number. That's from a virology class.
It was lethal.
98% lethal when they introduced it, and it's carried by mosquitoes. So mosquitoes bite a rabbit, and they bite another rabbit. A mosquito could bite you. You can get infected with it. You won't have any symptoms or signs, right? Like everything. Remember, you're always being exposed to viruses. That's something that they're around you everywhere. So initially, it was 98% lethal, and after the first wet season, it bloomed and took over the whole and wiped out 98% of the rabbits. But then rabbits would reproduce and do what they do, and then all of a sudden it came down. The number started dropping and dropping and dropping and dropping. And so the virus was the virus was resultingly keeping the rabbits alive longer, so they have a better chance of getting bitten by a mosquito and then spreading it to another rabbit. So it ended up, those are actually, the results are being changed now. In the last couple of years, they've noticed the virus is resurging back to its more lethal levels. So it can switch right back too, if it finds a way to do that where it's still evolutionarily successful. But the same case is herpes, right? I mean, herpes has been with humans longer than we've been humans. 230 million years ago, the herpes viridae family came into humans. So,
and to clarify, you studied this, right?
Yeah,
actually, you did research on this.
Yeah, I used to work on cytomegaloes and herpes. It was in an HIV lab. Got to wear all the fun suits and do all that fun stuff.
Hope so.
Yeah, no, it was a very fun time. But the herpes has evolved to a way that it's almost harmless for humans. You get a wound here, it's not too bad in terms of humans. I mean, I think anyone on our planet would rather probably get infected with herpes than they would this, COVID-19, get the symptoms of COVID-19, because it's been around so long. It can live with you your whole life, and it can spread to as many people as it can. That's really a well-evolved virus that's kind of been in there. Same thing we can say about HIV. There's a number of different viruses that have done it. On the other side, every rule, there's a counterexample. Some viruses have actually grown to be more lethal. So that's the grain of salt with that.
Right. I mean, if I ask my friends now, what would you rather have, herpes or COVID-19? I'm pretty sure most of them would say COVID-19, unfortunately.
Yeah. They probably most of them have herpes, but yeah, I'm not saying anything about your friends, Dave.
Yeah, there was a funny thing. Right when I got back to the States, I got back last year, and right when I got back, there was something like 24% increase in herpes in Coachella. I was like, well, thank God I don't listen to that kind of music very often.
You're
But
right.
So speaking of long-term damages, right? Some of them evolved to be very harmful, like this rabbit one that eventually kills them. But it evolved to be less harmful, and now it's resurging back. What are some? I know it's so new, and we won't really know the effects of it until later. But based on possibly earlier statistics on the SARS-CoV-1 and what we have seen now with doctors all over the nation and all over the world, from what I understand, it has like the x-rays show like black almost holes in the lungs and stuff like that. It sounds really, really, really terrible, especially when you combine it with some other infection, side infection or blood infection that might occur after contracting the COVID-19. So what are what are we looking at for like long-term effects and damage on the body?
It's bad in the lungs. I mean, I guess if you listen to some of the people who have it, some of the stories, the burning lung, I guess, is the worst when you have this. It's unbearable. I mean, there's different people have it to different degrees, so it's tough to say what anyone's results are. It is being found now in neurons too. They're finding it's going into your CNS, your nerves, and it can cause long-term damage to your nerves. When you look at SARS, for example, SARS was a little worse in terms of symptoms, signs and symptoms than this. SARS killed 10%, 9, 10, depending upon the numbers.
Which
SARS
lowers its R naught, right, and its ability to spread, which is why it wasn't as, for
right on,
example.
right on. Yeah, exactly. SARS also wasn't contagious. And this is a tricky situation to talk about here. SARS had to be basically in its debilitating state to be very contagious, and it was very contagious, just like this virus. But you can walk. The difference between SARS and this is you can walk around for days and spread the virus without even having symptoms. You'll get symptoms later, or maybe you won't. That was not the case with SARS. When SARS came out, you had symptoms and you knew it. You were in the hospital. SARS put you under, and that was SARS's big Achilles heel, is that it hospitalized nearly all the people who were contagious, which is why we didn't have a bad outbreak. So it's really tough. They call it SARS-1 and this SARS-2 because they were both coronaviruses. So SARS, when you look at the results of the people who had SARS, and that's probably our closest analog. We don't have 10-year out studies right now on this virus because it's right now. But SARS shows, I think it's a
SARS. You mean COVID-1?
So yeah, SARS-1, the original
SARS-1,
SARS,
the
right?
original SARS.
SARS-1, the one that happened over a decade ago.
Right.
That virus, they've looked at long-term survivals of the people who had it, and an incredible percentage of them have diminished lung function. And I'm sure you've been seeing this, where the people have for the rest of their life, as far as people know, unless they really start healing sometime after 15 years from having the virus, is they have diminished lung function. So even these people who did survive it, they have lifelong damage to their lungs. There was a paper that came out 2 days ago. I wish I had the figures, but they gave 3-dimensional views, these renderings of the lung damage. It's horrific how much lung damage these people are suffering, and the majority of it's by their own immune system, it seems like.
Yeah. So we're talking long-term lung damage, permanent scarring, permanent ability for your lungs to actually do what they do and absorb and distribute oxygen to your body.
Yeah.
Whoa.
And imagine how many people are going to have that. It's not pretty.
Yeah, before the herd immunity, they said a U.S. UCSF, I think, was the first notes that I read about this, talking about 60%, upwards of 70% need to get it before it plateaus and before we actually see a possibility for the extinction of this kind of virus, which is really incredible. I heard another professor at USC talk about the same thing. They're going to need between 60 to 80% of people to be infected before this virus slows down. And as bad as it sounds, those 80% are going, or 70% are going to need to be infected to save the other 30. It sounds like, if not this thing, if your antibodies diminish in 3 years without all those people getting in before getting extinct, this could come for round 2.
Yeah.
And by then, if our lungs are in bad shape, who knows what's going to happen? This might increase.
So as I was saying, the goal right now is to slow the rate down, right? And we're just biding time. We're buying time. I don't know what expression that is. We're buying time, right? So the idea is there's a whole bunch of drugs that are coming out, these antivirals that are coming out, and some of them are showed some pretty good promise. They kind of fizzled out. Other ones are looking much better. But so the idea is we're just trying to keep the infection low enough that we don't kill a whole bunch of people by overloading the emergency rooms until an effective antiviral comes out, until we understand how the virus does what it does more, and lastly, until we get a vaccine. Even if a vaccine is 2 to 3 years, if enough people get that vaccine, you can wipe this virus out off the face of the earth. It might go back to its host, but then it has to make that jump all over again, which is very inefficient.
So I heard a really crazy, I can't even remember, I think it was a podcast I was listening to. They were talking, it was an immunologist doctor, I believe from either Stanford or again San Francisco, I'm not sure what it was, but they were actually talking about the actions our president took at the beginning of this virus. And everyone criticizes, obviously, hindsight is 2020, but everyone was criticizing his response to the virus. And what I understood and what I took away from this talk was, there needs to be, when you have an outbreak like this, a global outbreak, there needs to be some form of infection before you can actually start the quarantine of the virus. There needs to be some form of infection. And they were saying the hospitals need to reach around 50% of the hospital occupancy before you start, or before you're able to do a complete quarantine and shut everyone from going out to do anything. And this was because they need the people who are, you can't quarantine immediately and stop all flights coming in, everything coming in, because the problem is this kind of virus will last a year and a half, 2 years in the population. You can't just shut your country off from the world for 2 years. That'll devastate an economy. But so it was going to come, it was bound to come here anyways. So the idea is that you let your population get it, and then once hospitals are reaching 50% or approaching 50% occupied by critical cases, then you imply quarantine rules, and then you imply drastic quarantine rules when they reach around 75%. And then you have like a trickle effect of people coming in to fill those last 25% of the beds. But by then you also have people leaving, healed or recovered patients leaving as well. So you're able to fill the hospital up while people are still leaving and able to take care of this new influx. But you can't just cut them off immediately. That was my takeaway from it. And everyone seems to criticize, and we could all point fingers all we want and who it came from, who it didn't come from, but the fact is it's here now. What are your thoughts on that? I was also on that boat, like, why don't we just shut off from the world? But we have all seen what happens, right? You shut us down, and our economy hits its brakes, and people can't pay rent. And
yes, exactly.
I mean,
that's scary.
Yeah, I mean, what are we supposed to do?
Yeah, that could be worse than the virus. I mean, if you don't want the prevention to be worse than the disease, right? So if you shut down all of these people's source to money, the problem is a lot of people are getting fired right now too, and they're losing their health insurance. Things like that. People can't go to the hospitals right now because they don't have health insurance, and so and people are losing their jobs. People aren't having any money to buy food with. I mean, this is a serious thing. This is starvation. You got a chance of dying if you starve. You got a chance of dying with the virus. I mean, these people are choosing to take the virus, and it's a really difficult, I think this may be one of the most difficult problems a president has had to deal with in the last 100 years.
Yeah. Since the H1 or since the Spanish flu or whatever that outbreak was.
Since the Civil War, since any of it. I mean, this is a really difficult conundrum, like with no easy answer.
Yeah. So what are we doing? What kind of steps are we taking then to, you said you're working on with this company that Photon Systems has been around for 20 plus years. They do UV fluorescence and spectroscopy, normally for NASA and the Department of Defense. So what are the techniques, I guess, that we're using now to detect this virus? I heard a lot of people are getting turned away. We're running out of test kits. I mean, running out of test kits seems like that could be devastating on its own. So what kind of steps are we taking then to make sure we're able to test everybody and get everyone safe?
Yeah. Like in a lot of these countries that are being hit hard, I know Italy's one of them. And I know New York, I think, has started this now. You cannot get a test right now in some of these places unless you're ready to go on a ventilator, because they have to know if you have the virus, and they're saving their tests for only the ventilators. My wife's sister is a nurse, and she wanted to get tested because she's taking care of patients with COVID-19. She couldn't get tested. It was a fight. It's incredibly difficult. So the idea that what we're working on right now is we're working on a rapid test. So the tests in overall fall into two main categories. There's a nucleic acid based test, which you'll hear about as the PCR test, right? It's actually reverse transcriptase quantitative real-time PCR. And what they do is they just take they're looking for the RNA of the virus, and they're amplifying and amplifying and amplifying at 2, 4, 8, and then they're looking at the spectra of the light that can come through that solution, and you're able to tell how much RNA is amplified. And if you get a positive amplification, it's absolutely specific for that virus. It's a very specific test. It does have false positives and false negatives, but it's a very specific test. And if the RNA is just the right length, for pretty certain that that person has the RNA. The other type of test is an antibody test. We've figured out through years of research that you can use our body's same ability to fight viruses to detect viruses. So we basically, you take a viral epitope, which is a little piece of the virus, and you make a whole bunch of that because you have to sequence the virus to know this, and you inject it into the ascites of mice, which is the stomach. And then the mice will pump out antibodies and cell lines that you can grab the antibodies from, and they'll just make tons of this antibody. The B cells turn into plasma cells, and they pump out. This is an amazing fact. In your body and in my body, our B cells produce 2,000 antibody molecules per second. Each B cell is making 2,000 a second when it's fighting a disease. That's a crazy number to think about, right? So yeah, that's how you.
How are we not winning? What are these viruses pumping out, man?
Well, you can have 1 million virus particles per milliliter of your blood. I mean, these things are small. They fit so well in there.
Is that by the time that your body starts pumping out these antibodies at 2,000?
Yeah.
Would you say 2,000 a second?
2,000 a second. That's one B
Jeez.
cell. And B cells have undergone replication. You might have a tremendous number of these also. B cells pumping out. Yeah. So
we should be winning, man. These viruses, what the our B cells are kicking ass.
We do something. So every time we try to figure a way out, they figure a way out. So
what are we doing then? How are we gonna, like you mentioned it before, that we're having this problem in Italy. I just got off the phone today actually with a friend of mine from Italy. He says it's really absolutely devastating what's happening. What we see, to give you an example, and I will talk to this guy, he's my friend Davi. I'll talk to him again on Tuesday, so in a couple of days, and he'll be on this podcast as well, so we'll talk a little bit about that. But he was saying that these numbers aren't even right. He says sirens are going off every day, all day, coming to pick up dead people, take them to the hospitals. It's really, really, it was so hard to hear, and it sounded like it was almost natural for him to say. So what are we doing then to make sure that we have sufficient amount of testing, make sure we're not turning people away, because this increases, as we mentioned, the R naught, right? If someone's sick and you can't get them tested, they're just going to go home and infect 2 or 3 more people. So what are you guys doing in particular to combat this?
Well, actually, the device right to my left is what I'm working on at home, because we have to work on it at home because we're quarantined from our work right
Good,
now.
good.
Thank you. So what we're doing is we're making a real-time viral test, and it's basically the testing process itself takes about 1 second, and it's a fluorescence-based measurement, and we can get a result back in about 1 second. Total with the nose. So what we do is we take a nasal swab. We wipe it on a surface and we test that surface, and we can actually detect the actual virus particles themselves. And we could do some pretty fancy chemometrics, and we can do classification, say, this is, this virus or that virus. And we do it very, very well. So we're trying to then deploy these in hospitals as a pre-screen. One is going to a hospital in Germany, I think Tuesday next week, and then one over here in Pasadena. We're sending these units out to begin their training. They have to go through training before they can get in there, and then we have to release the data to the FDA. But so the idea is this is used in 2 different scenarios. Let's call it the pandemic scenario, where you have the pandemic has started. In order to do the other 2 tests, you need a viral sequence to do the PCR. So a lab has to bring the virus in and sequence it. That can take a lot of time. And then the antibody thing that I was telling you about, how you make an actual antibody, that takes 6 months. That takes a very long time, a couple months to 6 months, depending upon how well the mice produce the antibody and make the B cells. But so what we're saying is we don't need that much time. We basically can set up a general virus screen to be put inside of all. Am I talking about pandemic or non-pandemic? Let's talk about pandemic first. So let's say the pandemic is already undergoing like this one, and you have a limited number of tests. Well, what you do is it's a triaging procedure. Right now, the triaging procedure that the hospital are using is, are you in contact with somebody with COVID-19? Do you have basically fever? If you check off all the signs and symptoms, and if you're in a serious risk, they then test you. Well, what this thing's goal is that it replaces a lot of that triaging. They say, okay, here, come over and swab this. It just takes a second. It uses a swab. There's no buffers. There's no reagents. There's no anything for it. The only disposable is a really long, horrible, uncomfortable swab. And you swab it, you scan it. Within 5 minutes, the results come back, whether or not you have. It's not a clinical diagnosis right now, but whether or not you have a large virus population in your nose that looks like SARS-CoV-2. And if you do that, then they say, okay, now we can waste that test on you because you've shown that you have a tremendous viral load inside of your sinuses or in your mouth, and then it's justified at this point. So it saves tests, because it's really a shame when you can't even test people who are getting ready to go on ventilators. It's a shame the situation that's happening right now.
And that's what's happening. They're turning people away at the front door that are having issues already, and they just don't have the test. They can't test them, and they can't take them. So they're turning them away, and they're going home and dying at home.
Yeah. The other thing that's really cool is this can also be used in non-pandemic situations. So they just put it inside of the hospitals. That's what more of the hospitals and more of the workplaces are wanting to see right now, is you put this inside of a workplace or inside of a hospital, and then people come in before they even are admitted to the hospital. You swab their nose, you scan it, you can see high viral load. It doesn't just detect SARS-CoV-2, it detects influenza and a whole bunch of other viruses, and even viruses we don't know about yet. So if you start to see an emerging group of viruses in an area where a bunch of people are coming in and they just have a tremendous amount of viruses and viral load inside of it's called sputum. It's a terrible word, but it's basically the snot in your nose. You can actually head off one of these pandemics with this type of system. And that's actually what the bigger push right now for this is this it's called surveillance. It's viral surveillance. It's a pretty cool concept. And just for people who are thinking this is kind of a quack or whatever, this is basically the same technology that's going up on the Mars rover. So the reason this technology was selected for the Mars rover is because it works and it's amazing, and it won out all the other bids. This is a great technology.
So there are viruses on Mars as well. Is that what we're going up to? So it's going up to Mars to it sounds like scan for biological stuff, and then what do you guys need here? You said it's going to Auckland or going to Germany.
Hmm.
What are you looking to do here with it? Where are we trying to get it? Are you trying to get it into hospitals? What do you need from, I mean, talk to the people. What are you guys looking for? What
Yeah,
can they
if
help
you
you with?
have contacts at hospitals, please get them in touch with us. We have, we're talking to UCLA right now. We're talking to Pasadena. We're talking to a bunch of hospitals foreign, like in Germany and Switzerland. You know, we need contacts at hospitals, people who can get us in there, because it's all about data right now. It's if we get enough data quick enough, we can get this in to help this pandemic. And we could put it in your workplace, you know, and monitor when people start going back to work. We can monitor viral loads and make sure nobody's coming into workplaces with a virus. So, or
Yeah.
with viral.
What I've seen lately is they got the temperature things, but what it sounds like is you're contagious and you're spreading the virus before you have a
Mm-hmm.
temperature. So the moment you have a dry throat, the moment you have any kind of symptom, and um, before they can push you away and say, "Hey, we just don't have the test," you can go ahead and try this out and see if you have anything, and if you do, you can go get the real test. So that's an incredible piece of technology. Um, I want to touch a little bit on vaccines. We mentioned it. How long are we looking? How long does it take to have a vaccine for this? I mean, the rats build up these B cells in 6 months. So what are we looking at for a vaccine? What do you think?
It's just a different process, but vaccines take a while. I think the average vaccine takes 10 years to go through. 10 years. I mean, clinical trials are strict. I mean, the thing is
We're
that,
doomed, man. We're
you know,
doomed.
you can sometimes if you make the wrong type of vaccine, you can actually make the infection worse. Some Australians, again, not to beat on Australians, but what they
Man,
did
they
is
got cane toads there that they introduced to try to eat these little flies, and now the flies just went up the cane. The frogs can't even get them there. The rabbits, they introduced the snakes to eat the rabbits. Now there's a snake population. What's going on, Australia? What's going on? What's going on, guys?
yeah, no kidding. So, anyways, they you can make a virus worse if the wrong thing happens in the vaccine. You don't want that to happen. You want to immobilize. Oftentimes when the antibody binds, the antibody's like a Y, and it binds to the virion shell. What you want to do is immobilize the virus. You want to make it so it can't dock, because otherwise your T cells can come along or your macrophages can come along and try to eat this, because it gets recognized by the antibody, and the virus then attacks the T cell and goes inside. That's the problem with HIV. A lot of people are wondering, why do we have an HIV vaccine right now? Uh, HIV's been around for a very long time. Maybe there is one. I haven't seen it. I haven't really kept up on HIV in the last 5 or so years. But you can actually make things worse with a vaccine. So it has to be done right. Um,
So we're looking at herd immunity is the only option. It sounds like at this point.
well, a
And
lot of
what
people have what's
and
called the Stafford Act. So the Stafford Act is basically, it kind of bypasses a lot of normal regulations. That's the reason we're allowed to get into the hospitals with this, even though it's not an FDA approved device. A lot like these, we're able to test all kinds of drugs on people to find out what drugs are effective, even though they haven't been sent through clinical trials right now, because these people are basically going to die. So they're basically guinea pigs, and that's actually kind of a good thing. You want to test these drugs and
right,
see. One person dying can save millions of people. Um, so yeah,
I think there
Stafford
was
Act.
there was those people up in the doctors up in what was it, Seattle as well. It was like one of the first trials of a vaccine, and thank you to them for stepping up to the plate and trying it. I haven't followed up and seen what actually happened to it, but that's kind of what they did, right? They invented what they would hope to be a vaccine, either with inactive
Mhm.
a viral inactive piece of the virus or some antibodies with some kind of fighting principle towards the virus, and they injected it in themselves. So after this, I think I'll have to follow up with that and see exactly how that's going, but I really hope that they're doing well. That's a noble thing to do, incredible thing to do. And same with you guys. I mean, in taking this instrument into hospitals, building a database to hopefully save lives, sounds like we're all on the same page with that. We all want that to happen.
It's
um,
it's a scary thing, like what we're asking people to do. We have collaborators all over the world right now a lot of us are hunkered down at home and not going out occasionally, walking to parks, maintaining our social distancing, physical distance, whatever you want to call it, social distancing. But we're actually asking our collaborators to go into BSL level 2 and 3 facilities and handle hundreds and hundreds of samples a day that they know are positive for COVID-19. And so, a lot of us are staying at home and doing nothing, but there are some real heroes out there who are really trying to come to the answers and get things that can help people. And they need to be applauded, just like the nurses and the healthcare people who are taking care of these patients. It's noble.
It's really incredible, seriously. You see all these posts right on Facebook and everything, like nurses with the masks and stuff like that, and people are making jokes. This is not a joke. These people are true. These are our frontline people. It's almost a war, and more or less it's a war, and people are dying, and they're there in harm's way. So seriously, thank you to them, and thank you to the doctors and nurses around the world. Wow, incredible job. So as far as drugs go, vaccine obviously sounds like it's not out of the question. And thankfully for this Stanford Act and some other things like that, that people are able to step up to the plate and do something very brave and try to combat this with injecting themselves with hopefully what could be a vaccine for this thing. And who knows, maybe they'll get it right first shot, which would be incredible. But what are we looking at? So what drugs do we see working and what don't? I've heard vitamin C. And also elaborate on this a little bit more. What's going on with the inflammation drugs and that 10xing? I heard a podcast, and I think I sent it to you earlier, about a doctor who said vast majority of the patients in critical condition were taking Advil, were taking anti-inflammatory, stuff like that. So what's going on with that, and why should we avoid that?
That's about 3 questions there. So as for the drugs, so I think you said drugs, vitamins. I should write this down. Drugs, vitamin C, and then
just what's working and what's not.
Yeah. Okay. So again, we're in the fog of war right now. Like, the information is flooding in. It's an amazing time in science, but it's also a time you have to take something with a grain of salt. Now. Which one of these do I do? Do I tie into first? Let's do the vitamin C, right? So vitamin C is giving, I was reading up on some papers on vitamin C, and is giving some mixed results, right? Some things are saying it's positive, some things are saying it's negative. Vitamin C, it seems to be the kind of the rationale behind vitamin C is some Americans are deficient in vitamin C. And if they're deficient in vitamin C, they have a weaker immune system. They can't fight this particular virus as well as they should be. So a lot of the positives, the people who are, the vitamin C is very effective in helping viral clearance. They're vitamin C deficient, and that's where a lot of the positive results for vitamin C is. It's very mixed. There's some positive results and some negative results if you have a normal vitamin C intake and you aren't deficient in vitamin C. It's not certain whether that will help fight the virus or not. But if you think you might be vitamin C deficient, you should take some vitamin C and get your vitamin C levels back up. It's not really going to hurt anything. Vitamin C can lead to indigestion and a few other things if taken in a very large quantity. But I don't think vitamin C is a negative thing. Is that what you're reading? I haven't seen what you're seeing.
The thing I read, and I'll try to find it again and again, put it in the comments section below. But the thing I read was a vitamin C complex combated with some other antiviral medication. I don't have the medication as well.
Is it hydroxyquinones?
I don't want to say yes, and I don't want to say no, but it was vitamin C with something else. I'll pull the article back up. But are there any things that you've read that seem to be working, whether it be in the hospital or at home?
Yeah. Well, so initially these hydroxyquinones, these malarial drugs, the hydroxyquinones and the quinones, they seem to expedite viral load clearance. Basically, what they showed was in patients who had medium infection, medium moderate levels of infection, they tended to get better a little bit. They tended to get better faster with if they were taking these hydroxyquinones. The mechanism for that, I don't know. I don't think anybody really knows the mechanism for that. But so the extrapolation was, hey, if these moderate patients are having a better outcome, a better clinical outcome with taking hydroxyquinones, well, then the extreme patients might have an even better outcome. But unfortunately, that's just not holding up. It's a scientifically significant hastening of viral clearance, but it's not substantial. It's not a large. I think instead of 7 days, you get better in 9 if you already have if you're already there. This isn't like, oh, it makes you better. It just makes you get better a little bit faster. We'll take it. You know what I mean? Like us as the world, we should take any advance we have, and that's why they're trying to combine other things with the hydroxyquinones. And some people believe it's due to the cytokine storm. This can mitigate that. But that's a whole nother subject too. So we've talked about vitamin C. We've talked about some of the drugs. Oh, there's other drugs too. There's a uracil drug, a hydroxy. It's like a 4. I don't remember the name of it, but it's one of these uracil drugs. It's a hydroxyuracil.
Are these over the counter? Can you get these or no? I've seen some malaria pills you can take over the counter, but what?
Yeah. So you used to be able to buy malaria or anything over the counter. And there were some people who I think have lupus who are prescribed these as it is. So these people with lupus, they're actually being denied their prescription for their lupus drugs because of the potential rush for the people getting viruses or people, testing.
Guys, don't be going out there and going to your friends with lupus and taking their medication either.
Yeah. So the other one, the newest, coolest one is this 4-hydroxy citidine, and it seems to be having a substantial effect on reducing the severity of the symptoms. But again, that may go away. Like this hydroxy. Now it's based on the one I just said 2 minutes ago. The hydroxyquinones. When the hydroxyquinones came out, it was a big buzz. Everyone was really excited about it. And then you have this like disillusionment, and then it might come back and be somewhat effective. And they're probably seeing that with this hydroxycitidine.
Right. And what about the negative stuff? I've read, I think you told me first a couple weeks ago about the anti-inflammation research coming out of China, and now we have doctors here suggesting that majority of their patients actually that are in the hospital in critical condition were taking, or hopefully they're not still, were taking anti-inflammatory drugs, which is quite scary because the older population, right? A lot of them take these Advils once a day for hearts, better circulation, stuff like that. What have you read or heard about this anti-inflammation stuff?
Initially, what China released was they were again looking at these clinical studies of patients, similar to what you're talking about, where they looked at actual people who died, and they said that, hey, look, what we were doing is we were giving the people steroids, we were giving these people anti-inflammatories, and it was I forget the terminology they used, but it was antagonizing the symptoms or antagonizing the severity of the virus. It was, I can't remember the exact wording. It's kind of important. And so that was the initial studies out of China that were saying this. And then as it infected France, the health minister of France, I forget his name, Veron, maybe you've heard of him. I know you spent some time in France, quite a bit of time. He basically came out and said the same thing. Well, these NSAIDs, I don't remember if it's Tylenol or Advil, I forget which one it is, but the NSAIDs, these non-steroidal anti-inflammation type drugs, were resulting in an increased mortality in the patients who have the disease. That was holding up. Italy kind of said the same thing. Then Italy also came out and said the same thing. The problem is now there was no clinical data supporting that. And it was these, these people coming out and saying this. It was almost anecdotal evidence. And so what happened 2 days ago, which is kind of interesting, is that Science, which is one of the greatest magazines of all time, they came out and said basically guidance about the effects of ibuprofen should be based purely on merit or purely on scientific research and not on these anecdotal stories. So the bottom line is in the WHO, or no, this was the FDA immediately after that, also retracted their guidance on, basically the FDA came out immediately after that saying these NSAIDs, not taking these NSAIDs should not be avoided or something. It was like a double negative. But what they were saying is we're kind of backing off on the NSAIDs. And that's the most recent thing as of 2 days ago. So basically, I think that the NSAIDs, I was the one who was saying that was bad. You got to stay up with the data. And I'm saying now, I think it's less dangerous than people actually thought. And if your healthcare provider is recommending you take these NSAIDs, that should probably be followed over these anecdotal stories coming out of these countries.
Right.
You know what I mean? Everything right now, I think it's 50-50. I don't think, of course, if you're just taking ibuprofen, or I forget, if you're taking an NSAID for not under a doctor's prescription, I would stop it.
Yeah.
But if it's under a doctor's prescription,
if you need it, do it. If you don't, I mean, give yourself, even if it is an anecdotal story, I mean, give yourself the best shot you got at fighting this thing if you get it. In my advice, which take that with a grain of salt as well, but do what you can to fight this thing. Do what you can to give yourself the best fighting option. I know you, Mike, I know you're exercising every day or trying to, and you're eating right, and you're you don't smoke, but
sleep, man.
Sleep,
yeah. Most important thing.
Yeah, so I guess tell us what this is a good thing. Tell us what your advice would be based on what you have read to give yourself a fighting chance at fighting this thing, or what would you do?
I'm not a medical doctor.
Yes.
Okay, I'm saying that right now. My advice is my opinion, and I base it on science. That's what I do with most of my life. And so please, if your doctor's saying otherwise, do otherwise. I'm not a medical advisor. This is not medical advice. The most important thing to boost your immune system is sleep. Regular sleep. You can look these studies up, Dave. I'll give you some of these studies. They're breathtaking. So what some researchers did was there's other, as I said, there's a lot of other viruses out there. Rhinovirus, it causes the normal cold. So what some researchers did was they took a large panel of people and they infected some people with they infected all the people in the study with rhinovirus.
What a cruel study.
Yeah, no, people get paid for it, whatever, people want to do it. But what they did was they had a group of half of them, they had them maintaining an 8-hour sleep regimen every night, right? Every single night, consistent. And the other half, they only gave them 6 hours of sleep. So this had been going on for a few weeks, and then they infected everyone with rhinovirus, just cold. And what then they did is they then told the people who had the 6 hours of sleep, go get as much sleep as you want, right? Get caught up on sleep 10, 12 hours, get all caught up. That was after being challenged with the virus. So what they found was the people who were consistently getting 8 hours of sleep had a 10 times higher viral titer, 10 times higher. Wow. There's rarely in biology do you see something that's that pronounced with a single small change. And when you talk about what your goals, if you're going to get this virus are, you want to shift from the population that is requiring oxygenation and that is requiring a ventilator. You want to shift out of that population and into the population that doesn't require those, that has the more mild, quote, mild symptoms or asymptomatic. And one of the best ways to do that is, of course, increasing your viral titer. And the way you do that is from sleep. It is the single most important thing you can do right now is get a consistent 8 hours of sleep, expecting that you might get the virus. So I'm really trying to stick with that. It's hard with this program we're working on. I've been working 100-hour weeks trying to get this out. But I'm really trying to get my sleep and, of course, exercise, trying to lose weight, because as I was mentioning earlier, the ACE2 receptors in your fat cells, if you can reduce some of the virus in your system, and that's why maybe some of these NBA players are not having much symptoms. A large majority of them aren't having symptoms. They're probably due to their low fat content. So reduce your fat content. And exercising helps for a lot of cardiovascular reasons, and that's why this virus kills you, is from cardiovascular reasons. Mitigate your diabetes if you have diabetes. Really try to not eat that much sugar if you have diabetes, because diabetes is one of the biggest risk factors for this virus also. So that's what I'm trying to do. I try to get a little bit of cardio exercise in every day, try to eat healthy.
So if you want an excuse to stop smoking or to get on a diet, this is how's death for you. Just one more question that a couple people had asked me, and we touched on it before. I just want to try to be more clear about it, and this goes back to the herd immunity. There were some stories coming out of, I think there was 2 or 3 people in China, and I read the other day about a few people in Japan that supposedly had caught this virus twice. What's the likelihood they caught it twice, or is it more likely, that it was something else, that false positive, false negative, whatever it was?
Well, I mean, there's weird medical stuff that happens all the time. It's a fact. Some people are born with webbed toes. No offense if you're born with webbed toes, it's just fine. But some people are born with tails. There's weird medical anomalies. When you have a large enough population, you can get some weird medical anomalies. That doesn't mean that's a norm for the population. That being said, personally, from the stuff that I've read, it seems that there's false positives with test number 1, right? People can be asymptomatic also, and that we know the virus undergoes a serotypic change. So you kind of combine all of those things and says, okay, a person was sick, they got a little better, then they got sick again, right? That's a fairly normal thing with some viruses. So I don't think that they were infected a second time. I think that the virus, there's not a large enough pool of virus out there to have evolved like the H1 and the H2. So the same person can be challenged with a different looking virus than whatever their B cells were recognizing. So I don't think they were infected by a different virus. It's more probable that either they didn't mount a successful immune challenge to this. They didn't make the right amount of antibodies or the right type of antibodies in the right amount, and it just kind of came back. It could have moved from their intestines. If the intestinal infection is true, it could go through viremia, through the blood, up to the lungs, and then start expressing in the lungs as well. It's a really tough situation. I don't think people are getting infected twice. I think what is happening is they're clearing the virus, and then another pocket pops up in them, or their immune system changes in some way, and the virus can get a foothold and kind of come back.
Or it goes dormant or something like this. Someone mentioned an article about dormants. Yeah.
Sure.
All right. Anything else you would like to add? I'm going to be talking to people from around the world in this series, World Corona Chronicles. So make sure to subscribe it. Register the ding too, the notifications, because these are going to be coming in 2 or hopefully 3 times per week, staying on top of this and staying up with my friends from around the world. Mike, would you like to add anything else? I love what you're doing with the instrument. That sounds like you really save some lives. So I know you're sacrificing your sleep for that, and thank you for that. So anything else you'd like to add?
No, I really like to hear from people around the world. I think that's a really exciting idea to talk with how people from all around the world are doing this, giving your perspective of how you've traveled. So I'm excited about what you're doing. So.
For sure. Okay, next week is going to be Italy and Spain. 2 countries hit very hard from this. And yeah, I'm excited to see how their approach was, and you will see as well. Thanks again so much, Mike.
Thanks, Dave.